welcome everybody to our today's webinar. I'm really happy that you all could make it to join us to learn more about B-cell analysis. And I'm really happy to make this webinar with you today because, of course, as B-cell product manager, it's my pleasure to tell you more about this. And I would just quickly want to show you the agenda. So what is the plan for today? So first, we are giving an introduction in the B-cell function. So just like very shortly about challenges that you face when you want to analyze B-cells. Then we're going into the technologies that can help you to analyze B-cells. And then in the end, we will give you some examples, so application examples on human and mouse B-cell analysis. And also in the end, I will give you an example for B-cell sorting for drug discovery processes. In the beginning, I just want to shortly show you some fast facts about Miltenyi. I'm not sure how aware you are of like the company. So just like, some numbers here. So the company was founded in 1989. So you see it's more than 30 years of experience. And we are about 3,000 employees working worldwide. And we have about 50 nationalities in our staff, what we enjoy very much. We have employees in R&D from about 23%. And we have more than 20,000 scientific publications from us and from all our customers using our products. We have a product portfolio of about 17,000 products. We did more than 65,000 cell therapy procedures with our products. And that means that about 2,500 patients per year are treated with our cell products using our technologies. And from the sales side, so we do sell our products in 28 countries directly and in 73 countries through distributors. So that was it already like from the Miltenyi side. So now we're starting directly into the topic of the B cells. So just like, some fast facts that you all might know, as you probably are all B cell researchers, but just like, to catch up everyone. So we know that B cells are producing antibodies. So that's, of course, like their main effective function. So they have a memory function, and that's why also like B cells and antibodies are quite interesting in drug discovery processes. Of course, B cells also produce cytokines. They play a role in wound healing, in antigen presentation, and also the balance of Th1 and Th2 cells, also via cytokines. They play a role in dendritic cell regulation, in tumor immunity, and then also the co-stimulation of other immune cells. So you see, like B cells are quite important, and that's why I want to target this topic today. And now I'm going a bit deeper into the B cells, so B cell types. So there are actually two types of B cells. So we have the B1 and the B2 B cells. The B2 B cells are the follicular B cells, normally always just like called B cells, and they are the largest population of B cells, and they play the part in the adaptive immune system. So that's like the commonly known, always referred to B cells. But we also have the B1 cells that are B cells that are T cell independent, and they have more innate like immune response. So they play the same role as like the other B2 B cells, so they make antibodies and act as antigen-presenting cells, but they produce like more like natural antibodies, and they also participate in the maintenance of tissue homeostasis and also play a role in the immune defense against mucosal antigens. So that's like their most important thing. Their repertoire is like more polyspecific, so they also bind to microbial antigens, but also to self-antigens. And in this B1 type of B cells, we have two subsets, so the B1A cells and the B1B cells. So here we have an overview how the B1A and B1B cells look in mouse. And so you see the main differences between those two cell types are that the B1A cells express CD5 as a marker and B1B cells not. That's why it's also like, discussed to rename them, and there's like quite some discussion going around this because like this naming is rather confusing. But the rest of the markers, as you see like on the list here, is the same. But I will not go into detail here right now, but you can always like, check when you get the presentation later. So here's like also the development stages of mouse B cells. So you see there is a lot and there are a lot of markers. So it's like really like complicated to remember all of them and to find the right marker combination to really look into the cells. And when we now look here in the list of the human B cells, so you see there are actually like quite some differences to the mouse B cells. So for instance, like the CD10 as a marker is quite unique to several stages of development for the human B cells, and also that we have CD38 as a marker quite early in development of B cells, and also another marker that is unique-ish for or that is like a marker for memory B cells, activated B cells, is CD27. So this marker cannot be used as a memory marker like this in mouse, but for human this marker is working quite nicely. And this like leads us already to the challenges that you can face when you have B cell flow analysis. So it's really a bit difficult because we have very inconsistent and not really like classified subsets in human. We don't really have that much phenotypic markers that are really like consistent. So it's really complicated to identify them because like you don't really have well-defined populations. They have overlapping markers so that you have like some populations that are like, overlapping so that you have like larger heterogeneous populations, what makes it really difficult. And then also on top it comes, of course, first mouse B cells were studied, and then all this concept were just like, used for the humans as well. And so a lot of researchers started in the very beginning to use the very same markers for human and mouse, and as you saw before, it's just not the same. So it's really complicated sometimes, and then also it's that like, it's more that the B cells are defined by their surface markers instead of really their functionality to look into what are they really doing. So that makes it even more difficult. So that's why we're giving you, when you're working with like, the main or like the main subset and not really like specific extraordinary subsets of B cells, to really focus on the core strain markers and use subgating strategies to really identify your B cell populations. But this we will explain you later in the application part. Now I will hand over to Omi, who's going to talk about the technologies that will help you to do standardized and reproducible B cell analysis.
Thank you, Susanna. Hello, everyone. Welcome from my side as well. So I would like to introduce you to the tools that Miltenyi Biotec offers to help your B cell analysis more efficiently and also to enhance the reproducibility of your data. But before going to details on this topic, I would like to discuss about the sources of data variation when actually analyzing cells via flow cytometry. So while performing flow cytometry, probably many of you have experienced that your data varies a lot from experiment to experiment. And also there are studies that have shown a significant financial effect of such reproducibility problems on research. There are several factors which actually affect such variability in flow analysis. And although one of the big factors of this data variability is coming from the donor, so the biological material that one is using, And this obviously can be dependent on environmental or genetic factors. For example, in this study, it showed different percentage of immune cells in different samples based on the donor, which is obviously a common source of variability and very difficult to handle. However, there are also some technical source that also affects the variability in flow cytometry. For example, the reagent that you are using to stain your cells, and also the instrument that can also result in different variability source based on the manufacturing tolerance, operational tolerance, and so on. And of course, some variability also arises from the data analysis and the protocols that are being used. And in today's presentation, I would like to focus on some of this variability and how we try to solve these issues with our solution. So obviously, here at Miltenyi Biotec, our team also realized these possible variability sources and put a lot of great effort to identify and solve such reproducibility issues that comes from these sources. And I will introduce you a few of these tools that we have developed. So since 2008, actually our company has invested heavily on flow cytometry technology development and also brought pioneering reagents in the field. Of course, I don't have the time to go through all the details, but I will introduce a few of these tools that you might find helpful for your study. So first of all, I would like to start with the reagent, because this is also reported in many publications to be the biggest challenge faced in the field. And in the last years, actually, the reproducibility crisis was extensively investigated, and antibodies were actually considered as one of the key variables that contributes to variability in cell analysis. And there were many journals, including Nature, who have published articles highlighting the importance of reproducibility and also the call for advancement of technology, especially for the antibodies, to support the cell analysis to avoid such crises. And some of the commonly debated challenges are the lot-to-lot variation, the background noise that arises from antibodies, and also titration and control relevant issues, and sometimes also reported to have the not properly validated application. So Miltenyi Biotec actually pioneered the reaffinity antibodies. These are recombinantly generated antibodies in an in vitro expression system, and this assures a very high purity and standardized production and also strict QC steps. And in this antibody line, we have the Fc region of these antibodies are mutated to abolish the unspecific binding to the FcR gamma receptor. And therefore, this also allows you to have only single isotype control for multiple reagents and reduces time for that. The antibodies that we provide are also pre-titrated and validated for flow cytometry application. And if you remember, Susanne mentioned that we have 17,000 products, and among which you can see almost 50,000 of them are different fluorochrome conjugated antibodies. We also have a separate webinar available to explain the technology behind these reaffinity antibodies, and also a special webinar focusing how we validate our antibody with very intense steps that are done in production QC. If you are interested, you can go to our website and have a look into those webinars. Now, when it comes to the flow cytometer instruments, there are some common challenges that comes with the maintenance and complicated use, operator variability, and sometimes sensitivity and consistency. In general, to solve such problems, a high degree of automation can help to reduce the human error. And in Miltenyi Biotec, we offer MACSQuant Family flow cytometer that allows you to have full automation without compromising the data quality. We offer the flow cytometer with automated use that is very easy to maintain. For example, we have automated startup, shutdown, calibration QC, these steps available, and also to avoid the operator variability, there is possibility to label your samples automatically and also acquire them. And the data analysis, for automated data analysis, I will mention later. However, as I mentioned, we don't compromise the data quality. We really ensure that the carryover between sample to sample is really low, and we also offer absolute cell counting that is volumetric, so you don't need counting beads for that. And as an advanced feature, we also offer unibody optical bands. So basically, you can transfer the configuration of one flow cytometer to the other, and thereby can align multiple instruments. Maybe if you are working on drug discovery, this could be a very helpful feature for your cytometer. When it comes to the data analysis, as you can imagine, for flow data, it's always the gating that comes into play, depending on who is drawing the gate. Here is an example. I mean, this is maybe a little bit extreme, but you can imagine of a time when you see the gate of your data and you are not sure where to set the gate. And this is actually one of the most common variability that arises when analyzing flow data. And this results in lots of error, takes time, and yeah, the interpretation of data can also vary with that. And here, to avoid this manual gating related problem, we offer the express mode solution together with the MACSQuantify software that is integrated in our flow cytometer. And what these express modes are doing is basically a standardized algorithm-based data analysis platform. It simply helps you to automate your data analysis by defining the gate, where to set the gate. And thereby it allows you to standardize your data analysis, and you can keep a consistent data analysis from operator to operator, thereby ensuring reproducibility. And this is very easy and quickly can be integrated to the SOPs for validated automated analysis concept. I would just quickly want to show an example how it looks like. So we offer different express mode packages, and one of the packages for express mode includes, particularly that would be for your interest, is for B-cell quantification. So here in this express mode, you have the possibility to have an 8-color panel as displayed here. And you can quantify of different B-cell subset, for example, your activated B-cell, naive or memory B-cells. And with this automated express mode, it will give you this kind of plot. So what you will see is automated analysis of dot plots with the gating set automatically. And also you will find a table with all the statistics that include the count and the percentage of your population, and you can export easily this data as a PDF or Excel file that you can eventually follow up later. We actually, in our express mode, as off the shelf, we have 3 different packages available for B-cell. So for B-cell differentiation, B malignant cells, and B regulatory cells. So these 3 different express modes are available. All of them includes a color panel, but obviously this is a feature that is integrated with our MACSQuant flow cytometer. However, so this was a very summarized way to show you what we have to offer from the product perspective for your B-cell analysis. So together with our integrated instrumentary and software solutions, we ensure the reproducibility, standardization, and automation for your assay.
Yeah, thank you, Omi, for explaining us this. And now we will go into the application section. So I will going to show you now some examples, like first for human B-cell analysis. So here I have one of the panels that our R&D is regularly using. So here the data I'm going to show you is like from B cells that were purified using CD19 microbead kit human. And we stained the samples with 7 different recombinant reaffinity antibodies and 1 viability dye. So we have like here on the right side the list of the markers and antibodies we used. So CD19, CD20 for usually like to get on the B cells, IgD to check naive B cells, CD27, as I mentioned before, is a marker for memory B cells, CD38 for plasma blasts and plasma cells, also CD138 for the plasma cells, then CD86 as an activation marker, and MHC2 also as an activation marker. Now I'm going to show you how this actually looks like. So we started with this like multi-step gating strategy. So of course first cell debris, doublets and decked cells were excluded. So that's like always the first 3 steps. And then in the next step we already gated for the plasma cells. So this is like the gate up here. What is like CD38 high and CD138 positive. And you see the percentage of plasma cells usually in human blood is like pretty low, so it's around 0.01%. So that's why it's always like so hard to find them there, but that's how you gate them. And then in the next step, we gated on CD20 positive B cells. So we made like a nice gate here. So there you have like all your B cells in was like around 99% of the cells. And then we looked with the multi-step gating strategy to check this gate for switched memory B cells. That's the gate up here that are IgD negative and CD27 positive. Then we also have a non-switched memory B cells that are IgD positive and CD27 positive. Then we have naive B cells that are IgD positive and CD27 negative, of course. And then we have this double negative, or also called unconventional memory B cells that are IgD negative and also CD27 negative, and that's why they're in the gate down there. Now, when we want to look in a bit closer and see how's the activation status of these B cells, we always check for MHC2 to see if the B cells are expressing MHC2. So you really see nicely here that they're expressing MHC2. And also on top, we always stain for CD86 on top to really see if this is like also going to be positive or not. So you see like this isolated cells are not like activated, just like around like 1% of the cells are CD86 positive. And that's why we also on top like analyze MHC2, because sometimes it gives you like more information when you stain for both of the markers. So this is like, our general like B cell analysis panel, what we use in the lab at Miltenyi. And now I will show you an example for mouse B cells. So this is like a bit more complicated, so to say, because we use like a B1 and B2 panel. Because of course we can like use other samples than in mouse than for human. And that's why we have like this B1 panel, what's consistent of CD19 for the B cells again, IgD for the memory and the naive B cells, CD43, CD45 receptor, then CD5 to look into the B1A and B1B cells, and also IgM. And then the B2 panel, so in the second tube, so to say, we have CD19, IgD, CD23, CD45, CD21, 35, and also IgM, so slightly different than the B1 panel. And now I'm going to show you the application data on this. So we also have the very same starting point in the gating strategy. So we first exclude cell debris, doublets, and dead cells. So I'm not going to show you that again, so you already know this. And then you can gate for the B1 and the B2 cells. So the B1 cells, this gate here in pink, is CD19 high and CD45 receptor low to negative. That's why it appears here. And then also we have the B2 cell gate, what's also about 21% of the B cells, what is CD19 high and CD45 receptor positive. So these are the two gates. You can get first the B1 panel, but as it is the B1 panel, we want to look closer, of course, into the B1 cells. So we're using like the gate that I showed you before to look into the B1A and B1B cells by checking for the markers CD5 and CD43. So as you see here, most of the B cells are CD5 positive, so most of them are B1A cells, so more than 60%. And another around 8% are CD5 negative and CD43 positive, so these are the B1B cells. And then we go closer into the classification of the B2 cells. So now we use the B2 panel because like you can nicely analyze the B2 cells there. So here you see like the B2 gate that is like, up here. And here you can look more closely into it when you check for IgM and IgD to see if you have mature B cells or transitional B cells. You see here, up IgM positive and IgD positive are the mature B cells, and the transitional B cells down here was just only like about 10% of this B2 cells in the mouse splenocytes. And also you can then look closer into the subsets of the mature B cells. So we have marginal zone B cells that are CD21 positive and CD23 negative. So that's this gate up here. And then also you can check here for follicular B cells that are CD21 dim and also CD23 positive. So this gate over here. So you see like that almost 90% of this mature B cells are in the follicular zone of the spleen. Also, when you want to look closer into the transitional B cells, you can actually distinguish between 3 different stages of transitional B cells. So it's T1, T2, and T3 cells that you can distinguish by the markers CD23 and IgM. So the T1 cells are CD23 negative, IgM positive. The T2 cells are positive for both markers, and the T3 cells are CD23 positive and IgM negative. And now I'm going a bit more into another topic, so also like the flow sorting of B cells for the therapeutic antibody discovery with our gentle cell sorter with a closed cartridge system. It's also like a very interesting system. So here is our antibody discovery process that you start with like your sample material of interest. So either like human, blood, or mouse spleen. This depends on like, what you're interested in. If you have like, immunized mice or like maybe a patient like, in the cases now, you maybe want to look into corona-specific antibodies. And for this, we have like several kits where you can like isolate your B cells from different starting materials. So from human blood, we have the straight from products when you can directly isolate your B cells from the blood or from different blood products. And also we have isolation kits that isolate the B cells from PBMCs or also like from the mouse splenocytes. So there are like all the options you need for your research to isolate your B cells. And when you isolated your B cells, of course, you're going to use like the panels we suggested before to really see what antibodies, what B cells you're having, if you're really having what you wanted, how is the purity. Of course, you want to do a bit of QC with the MAX antibodies and the MACSQuant analyzer. And then the next step would be cultivation of your isolated B cells in the presence of antigen to really like push your antigen-specific B cells. For this like culture conditions, we have this nice B cell expansion kit. We have CD40 ligand multi-MEC kit, a lot of TLR ligands and cytokines that you can add and really use in the culture here. And then in the last step, before you really get your lead candidates, you do select the binders because you want to have your antigen-specific B cells, and that's you can do with the MACSQuant Tyto with fluorescence labeled antigen, what's like really nicely working. And then you can, of course, like clone recombinantly express your antibodies to go into functional testing, where we also have a lot of different products that you can use. So if you want to know more about this, I'm going to hold a webinar on this in September. So I'm really happy to welcome you all there. And so just like so that you know, like what we are talking about. So this is like, the MACSQuant Tyto data. So you really see that when you're starting from a little number of B cells from about like 4%, you really get like nicely sorted your 95% pure B cells. So this is like a example from PBMCs with a very high viability of about 99%. And the nice thing is on the system that is like really closed cartridge system was like super easy to start. The summary. So we really want to help you to improve your B cell flow analysis. And so we wanted to just like show you that we have recombinant antibodies for reproducible immunophenotyping of B cells. So I hope that got clear. We have automated flow cytometers equipped with B cell specific data analysis software that really helps you to analyze your B cells. And we have a system, so the MACSQuant Tyto, that helps you to really gently sort your B cells for any purposes. And also we have an expert team that supports and helps you to build your panels and select the right marker combinations for your research. So if you need help there, just like contact us, and we are helping you with this research. And so now I'm at the end right now, and I thank you very much for your attention.